greatest research. And so, so as Zia said, she was my PhD student, actually my second PhD student. So she took a chance on my lab as a new faculty. So I was super excited to have her in the lab. And she did some really cool work on understanding the role of metal reducing bacteria in releasing arsenic from sediments. And there was a lot of questions at the time about how that happened, and arsenic and groundwater. And so she really put the power of genetics to merge the field of geochemistry and genetics in a very unique and powerful way to solve the problem. So she did figure out through her thesis that microbes, in particular, this particular pathway caused this arsenic to come off the surface of soils. And so that was a huge discovery for our field. And so I was really, really ecstatic about her results. And she published some really nice papers from her thesis. So before that, she was a master's student and Professor Tina Salmassi's lab at Cal State University, Los Angeles, and then CO2 and looked at microbial diversity. And interestingly, Tina Salmassi was a PhD student at Caltech, and I was a postdoc at the time at Caltech. And so circumstantially, we knew each other through Professor Salmassi. So yeah, and then before that, she was got her bachelor's degree in microbiology and molecular genetics at UCLA. And so you can sort of see the trend of molecular biology and geosciences. So, and so then she she received numerous awards and awesome fellowships through UC Santa Cruz, but one in particular was the UC Toxic Substance Research and Training Program. And at the time that funded a lot of grad students in the toxicology field in California. And since that program's been no longer functioning, but so she gave a keynote talk at the year later at their symposium. So that was a really nice fellow. And then, so I'm kind of jumping around. So after she got her PhD, she went to University of Bremen in Germany and to hone her skills on understanding how iron-reducing bacteria and marine cells. A lot of questions about that. So she was in the lab of Professor Michael Friedrich, and she got several really prestigious fellowships. One was the National Science Foundation international postdoc fellowship, and then to fund her time there at University of Bremen. And then she completed that postdoc and joined Professor Stefan Kramer's lab at University of Vienna. So, you know, nice place, right? To study archaea and their role in copper acquisition and so that's her latest work here. And so she's published some really nice papers, has a recent paper on, and it's an amazing paper. I looked through it, and the gene terms, I'm sure, well, I don't want to give it away, but
you're trying to scoop her.
So right, yeah, exactly. So really nice work. And so she received another really prestigious fellowship, the Marie Curie fellowship, to do work in Professor Kramer's lab. And so and then she's finishing up that right now, and I'm sure she'll talk about it, but she's taking a position in Switzerland to work in this with some engineers and scientists to work on bioenergy projects and fungal biofuel cells. So I just learned that just recently through her. So it's a really, really cool work. And so, yeah, I mean, I go on. Oh, and the other thing I want to mention, she received the Fulbright International Fellowship. So she really, did really well with funding her postdoc and published some really nice papers. And I'm just super ecstatic that she's here. And she's going to tell us about her work in archaea and iron-reduced iron and copper. So I'll stop talking and let Dr. Reyes take it away.
Thank you so much, Chad.
Thank you.
Just before Carolina starts up, for everyone that's here that is not familiar with the format of the BD2K Summer Talks, we always ask our speakers to talk a little bit about their path to their career. So that you may be surprised if it's, it won't be a 100% science talk, but the science will definitely be the central feature there. But you'll also get to hear all the cool about Carolina's path to where she's at now. So take it away, Carolina. Thank you.
Thanks. Okay, can everyone see my screen?
I can.
Yeah.
Okay, so before I get into the science part of the talk, I actually wanted to give a little more background about myself and maybe talk a little bit more in detail about some of the experiences that Chad talked about. So in this slide, I want to show you a little bit about where I grew up. So I spent my whole life into my postdoc. So originally I was born in Los Angeles, but most of my youth was spent in Santa Monica. So I went there for, elementary school, middle school, and high school. And my parents are pictured at the bottom, and they've actually been, the reason why I consider myself to be very hardworking. I have family that still lives in Los Angeles, and there's a picture of my family in LA next to one of my parents. But I also have family in Mexico. So throughout the years, I've tried to divide myself between, my family and LA. And actually, that's just a small bit of my family. My family is actually includes cousins and aunts, which I couldn't fit on this PowerPoint because there's so many. So my family has been also instrumental in supporting me throughout my career. So Chad talked a little bit about the places where I studied and I worked. And he mentioned at UCLA was one of the places where I started out with my research, and actually being a part of a STEM program was very important during my time at UCLA because it allowed me to, for the first time, actually enter the world of research and find out what that was all about. And it also allowed me to meet for the very first time other researchers that looked like me. And they came to be really good colleagues and really good friends. So until now, I have this very positive experience. Oh yeah, something else that I wanted to mention was when I actually went to UCLA, I was deciding what to study there. I was considering between being an English major or a biology major because I love to write a lot, but thankfully I went for biology. And yeah, so after completing my bachelor's there, I took a little bit of time in between to try and figure out what I wanted to do next. So in between, I was working, and I was also taking post-baccalaureate courses at Cal State LA. And it was during my post-bac courses that I found out about the C.A.R.E program there, which is another STEM program. And they offered the opportunity to become involved in research once again. And it was through the C.A.R.E program that I met Dr. Tina Salmasi, and she was a young researcher starting her own lab as well. And just by talking to her, I was very much interested in her work in environmental microbiology. It sounded fun, it sounded exciting, and so I decided to, take this opportunity and apply. And thankfully, I got into the program. And it was actually at Cal State LA where it was the first time that I could be in an institution that was primarily composed of Hispanic and Latino students. And that was the first time that I was in this type of environment. And so for me, my time at Cal State LA was a very great experience because of this. So during the master's, I also was very encouraged by Dr. Salmasi to go out and explore the field of geobiology and geomicrobiology, which up until then I didn't really know. Within the context of my own research, I always saw it as environmental microbiology, but there's actually a bigger component to that, which included geobiology. And so I applied to a program at USC where international students from Europe, but also from other parts of the US could join, and we could learn from geologists about geochemistry and geology and how that could actually intersect with the field of microbiology. And it was through this program that I met for the first time students coming from institutions and universities which were among the top ranking institutions and universities in the world. And it was the first time that I had thought about or considered that this could be an option. Coincidentally, that was also the program where I met my future husband. So that was also something that came out of that, the program. So following my time then at Cal State, I was once again motivated and also encouraged by Dr. Salmasi to consider pursuing a PhD. So actually, when I was doing my master's, the whole time I was thinking, okay, well, after doing the master's, I'm probably going into biotechnology or, you know, doing some research as part of a company job. But as I pursued my master's and I got more interested in the science and in the topic, I realized I wanted to continue my studies and become more experienced. And so when Tina mentioned that one of her colleagues was actually starting a lab at UC Santa Cruz, I was really excited about the opportunity of going there and pursuing a PhD. And so after much encouragement from Dr. Salmasi, I applied and I was accepted into the PhD program as part of the Microbiology and Environmental Toxicology Department, or MEDICS, as it came to be known. And of course, I'm really happy to have, you know, had this opportunity and to have been a part of Professor Saltykov's group. And I'm thankful that he took a chance on you as well. So at UCSD, I received a lot of support from the STEM diversity programs. They were the people that I turned to in times of difficulty or struggles during the PhD. So I'm very thankful to programs for being there and showing me that I will be experienced. If I fast forward now to almost to the end of my PhD, I had 2 years left, and I was thinking I really want to go abroad, partially, of course, because my future husband was there, but also because I wanted the opportunity to see what it would be like to study at a research institute outside the US. And so I started to think about where I wanted to take my research, how that could fit possibly into some of the groups that were in Europe. And it took me a year to finally find a host that was willing to host me as I got funding to go there. And it took me another year of writing and applying to these various research programs and hoping that I would get funded to go to Germany, and specifically to the University of Bremen. And thankfully this worked out, and I have to say I'm really, really thankful forever to the department because at the time they always had us do a mock exam where we would present a proposal and we would have to defend it as part of the PhD. And I have to say that was really instrumental in preparing me for applying to these various programs. And also the grant writing courses that were offered at the time by the department, they were also really important in preparing me to be in this kind of mindset of how do you develop a project. And I'll actually talk a little bit about this process of developing a project when one tries to acquire funding. But I'll talk about that a little later. So during this postdoc, that's when things got real after the PhD, because it was the first time that I had to manage a research project on my own without supervision. And this was with funding from the US, so I had to figure out how would I handle the finances to this project from Europe. That was a bit challenging. And in addition to that, when I arrived there, I learned that the collaboration I had set up had fallen through. And that's because Cruz, that was supposed to go out for sample collection, was not funded. And so the Cruz that was supposed to happen didn't happen. And I had to figure out quickly who else I could collaborate, who could acquire samples to allow me to do the project. So that was a very challenging time as part of that postdoc. But thankfully, that all worked out in the end. In between my first and second postdoc, then I was trying to wrap up the project. I was writing the first paper. I was looking for a new host at the University of Vienna, because by then I had gotten married, and my husband, being a scientist himself, he got appointed a research position, so I plan to move there as well. And that was also quite a challenging time, trying to basically join a new academic system in a new country once more. And I'll talk about those challenges as well. So by the time I moved to Vienna, I had figured out that I wanted to work in a geochemistry lab. This would be the first time that I would switch working in a traditional microbiology lab to something completely different. And I made that choice because I wanted to challenge myself. I wasn't challenged enough already, but I really wanted that opportunity to see how it is that geochemists worked and how they thought. And also, that means during my career in environmental microbiology and in geomicrobiology, I was always on the other side. And so it was exciting to have the opportunity to now immerse myself into a different environment and see my research, but from a different perspective. So by the time I moved to Vienna, I had applied to several research funding agencies, and I'll talk about that a little bit later too, but most of that actually fell through. I wasn't able to secure funding, so I spent a year looking for funding, finishing off manuscripts, but thankfully being a part of a department during this time really helped to keep the motivation high. And so I'll talk about this a little bit later, and now I'm going to move to a very personal part of the presentation. Well, I will talk about some of the challenges that I have faced, also living abroad. So some of the challenges that I faced while I was in the US, one was becoming fluent in English. So English was not my first language. Spanish was my mother tongue. And so for many years, up until almost middle school, I was in bilingual or dual language courses where I was working to become fluent in English. Something else that was challenging while living in the US was identifying as being Mexican, but also as being American. And there were times when I felt like a lot of the Mexican people that I encountered felt I wasn't Mexican enough. And there were other times when I encountered a lot of American people that didn't think I was American enough. And so it was always this conflict in me trying to prove that, yes, I am really Mexican, and yes, I am really American. I'm a mixture of both. And something else that was also challenging was growing up with discrimination while I was growing up. So there was a time in one of my classes where actually a guidance counselor stood up and in front of everyone told me, you know, there's no way that you're going to become a scientist, because as far as I know, no one in your family is involved in science. Thankfully, I had good colleagues who pointed out to him, you know, this is not acceptable. And so I had people standing up for myself. I was often told at university as well that the only reason I was there was because of affirmative action and not because I actually was smart enough to be there. But of course, I knew that not to be true. There are times as well where strangers would say, you know, go back to your country based on how it looks. You know, but despite facing all these challenges in the US, what really helped me overcome these challenges was having the love and support of family and friends who believed in my dreams and who encouraged me along the way. And I'm really, really thankful also for being a part of the STEM programs, because that was a very inclusive community, and I also felt very supported. So now I want to talk about some of the challenges I faced while abroad. So I chose to go to a German-speaking country, well, two of them, and a third one, which I will integrate into. So the biggest challenge was trying to integrate into these countries. So that meant learning German, trying to make time for that during a time when I should have been fully focused on the research, but of course I wanted to fit into this new society. Trying to understand cultural differences. So at the first time that I moved to Germany, it was in northern Germany, and so it was very, very different culture, very reserved, I would say, very conservative society. And so I had to adapt my behavior in order to fit in. So initially, I remember people being shocked by how loud I was or how much I was laughing. And so I had to learn to adapt my behavior in a way that I would feel, more accepted. There was also the issue of becoming accustomed to a new academic system. So in a lot of German-speaking countries, there's a hierarchy to the system, much more than I encountered in the US. So I also had a challenging time adjusting to that. And another challenge was using my time efficiently during times where I had no funding. So that meant networking with as many people as I could in various institutes, at conferences, volunteering at organizations so that I would stay involved in outreach, for example, and also to try and get to know what other groups were doing. And of course, always writing grants, writing manuscripts. And what continues to be a challenge is more so now, of course, in the time of COVID, is maintaining contact with family and friends in the US. So Chad mentioned a little bit about my research interests, and I kind of give you an overview here of what those topics were. So as Chad mentioned, during my PhD, we're in Shewanella ANA-3, which is a bacterium, and studying the iron reduction pathway in this organism. Following my PhD, I was interested in now looking at the environment to try and understand how microorganisms that carry out this pathway actually behave in the environment. And so that was the topic of my postdoc in Bremen. Following my postdoc in Bremen, I got involved in a project to look at copper uptake by archaea, which I will talk about today. But I've also had the chance to collaborate with the geology department looking at fungal quartz weathering. So I have quite a broad scope of research interests. So I said earlier in the presentation that I would talk a little bit about what it was like to obtain funding for my second postdoc. I know many of you might find yourself in this situation at some point. So my original idea was actually to work on a project looking at iron uptake and mineral dissolution in fungi. And this is the application that I was submitting to funding agencies during a period of 2 years with no luck. So after the third rejection, I decided to switch topics. And that was because an opportunity arose to develop a project with 2 different departments. In one department was a geochemistry department with Stefan Kramer, and the other department was with an archaeon department. And so it was with these 2 collaborations that I was then able to start to form the topic for my second postdoc. So, and how did the project idea arise? Well, there were previous studies going all the way back to 2004, when for the first time there was a molecule which is called methanobac. It's a type of ligand that can bind copper very tightly. And this methanobactin was the first of its kind, and it was observed in bacteria that's able to use methane. And so out of this research, there was this whole new field that opened up into methanobactin research and also into copper acquisition research with respect to bacteria. And by 2013, there was a research group that started looking into archaea. And so the first study that was published along these lines was looking at the ammonia-oxidizing archaeon, Nitrososphaera maritimus. And it was really the first paper to tackle this concept of copper uptake and requirement by an archaeon. And it was because of this study that the idea started to form within these 2 groups, the geochemistry group and the archaeal group in Vienna, how would other types of archaea respond to copper limitation? How do they go about capturing copper? So all these wonderful ideas were coming up, but they needed someone to write the proposal, plan the experiments, and make it a reality. And so in I came, and I learned about the Marie Curie action program, where it was possible to come in from a different country to try and raise funds to work in another country. And so it took me a year to write and develop this proposal to address the question of copper acquisition by archaea. And it was another year later until I heard a positive outcome, thankfully, from the organization. And then I had 2 years to try and accomplish all the goals that we had set for the project. So now I want to talk a little bit about what it took in order to put together this proposal. And this holds true for not only developing this project, but also previous projects and future projects. Development is determining what your aims and hypotheses are going to be. What is the question behind the research that you're trying to address? Once you have these 2 points, then the next step is trying to figure out what experiments are you going to need to address those aims. You start to think about who your team members will be, what kinds of specialities they have to have in order to make the project successful. And also you have to think about how much money do you need for this project within the time span that you have, and how are you going to break up the different experiments and milestones within this time frame. So these were all the things that I consistently kept in mind and will consistently keep in mind with future research. And now we get to the topic then of copper uptake. And so the question here is, why is copper physiologically important? So we know from previous work that copper is quite important as a cofactor in enzymes, and this is true for bacteria, eukaryotes, and archaea. That's why I have this tree of life on the right-hand side. And copper, when it forms a cofactor or a complex with an enzyme, can shuttle electrons. So it's capable of being reduced and it's capable of being oxidized. And I have 2 of the redox states of copper at the bottom here. And so where copper becomes important in terms of nitrogen cycling is that we know that there are various microorganisms that can carry out nitrification, which is part of the nitrogen cycle, and it's shown in green. And we know there are other organisms that can carry out denitrification steps, and that's shown in yellow. And we know that there are several steps in which you have enzymes that rely on copper in order to carry out those steps as part of the nitrogen cycle. And so one of those steps is the ammonia-oxidation step, which is a part of this nitrification step. And so, you know, microorganisms are very critical in maintaining this global nitrogen cycle on our Earth. And so these organisms be an important part of this global cycle. And specifically for this particular study then, we decided to use an archaea called Nitrososphaera viennensis, or N-viennensis for short, that is known to oxidize ammonia to nitrite. Now we know from previous research that it relies on the copper-containing ammonia monooxygenase in order to oxidize ammonia to nitrite. What isn't known, but it's hypothesized, is that it also requires the second enzyme, hydroxylamine dehydrogenase, in order to carry out the whole metabolic step. And so in this slide here, what we have depicted then is the hypothetical pathway, which is based on a lot of research that was performed previously. And so, as I mentioned before, we know for sure that there is a copper-bearing monooxygenase, which helps to transform ammonia to hydroxylamine. What's hypothesized is that there probably is an additional copper-containing enzyme, which will then convert the hydroxylamine to nitrite. And in between, we have molecules that function as electron shuttles, which will pass electrons to these hypothesized proteins as part of the electron transport chain, enabling this organism then to transform ammonia to nitrite. And so the first aim that I want to talk about involves looking at the effect that copper limitation had on the physiology of viennensis. And our hypothesis was that copper limiting conditions would inhibit the ammonia oxidation pathways of viennensis. So our experimental approach involved using a chelator called TITA. Now, TITA is a molecule that binds copper-2 very tightly in order to form the molecule structure pictured on the right. And so as part of the experiment, we wanted to use this copper chelator that was specific to copper-2 as a way to limit the amount of copper that would be bioavailable to viennensis. And so the idea was to set up experiments in which the organism was grown in its cultivation medium, which is in this case is freshwater medium, in the presence of this chelator. And we would add different concentrations of copper to try to reach a certain threshold of copper that would be available to the organism. And at the same time, we would set up control experiments where the organism would not be exposed to the TITA, and then compare the two types of responses. So the method that we used to go about this, initially we used a geochemical modeling program called C.A.R.E. And this modeling program allows you to play around with the parameters based on what kinds of ions are in solution. So in this case, in our medium. And this would predict or estimate that copper-2 plus would in theory be available to the organism under the cultivation conditions that we would use. Once we have these estimates, we could then actually set up the wet lab experiments where we would add TITA and copper to the medium to achieve these various free copper-2 plus concentrations. So we ended up growing these cultures and measuring the amount of nitrite that was produced from the reaction, and also the amount of ammonia that was consumed. And for the experiments themselves, we used a consistent amount of TITA, but we varied the amount of copper that would be available to the culture. And the first series of experiments that I performed were to test whether or not the TITA itself would be toxic the organism, because if it was, then we would not be able to use them in the actual limitation experiments. So initially what we did was to compare a copper replete culture in which no theta is available, just the normal amount of copper that is in the medium. And in the other case, we added the theta chelator to the cultivation media and then compounded by adding copper, so that at the end, we estimated that the levels of copper would be the same between the two. And so again, the idea here was to test whether theta by itself would somehow be toxic to the organism. So shown here are the results from that first experiment, or series of experiments. So these are all biological replicates and multiple experiments. On the y-axis, you have nitrite production, and on the x, the number of days. And what you can see is that both the copper replete and the copper theta cultures were producing relatively similar concentrations of nitrite. And so that was really a really good result for us, because it told us we could go ahead and use theta as a chelator in order to set up the copper limitation experiments. So I kind of want to pause here in between, and I will do that throughout most of the presentation to address challenges that I face along the way, and also to address things I learned as part of some of these experiments. So overall, the most challenging aspect of this project was trying to control for contamination. And that was not only controlling for kinds of copper and trying to prevent copper getting into these growth mediums, but also controlling for other organisms, so bacteria from getting into these archaea cultures. And that meant learning to work in a clean lab environment, and learning to work mostly with plasticware, because any glassware would for sure contain high levels of metals. Learning to cultivate a new organism, that was also very challenging. So I spent the first 3 months of this postdoc learning how to handle the organism, how to cultivate it, how to measure for nitrite and ammonia production. And along the way, there were moments where, oh no, you know, my cultures are dying off, or copper levels are going up, or having to restart things from the beginning. So these kinds of challenges don't stop after the PhD. They continue throughout your whole research career here as a scientist. And so kind of to give you a sense of the timeline, so I started the project in June. And at the same time, I started to lecture my own course for the first time. And between that time and the time I got the first set of results I just showed you, all this time had passed, and I was writing in between, starting to network and find more collaborators, and also mentoring students. So some of the lessons I learned initially was that setting up 2 microbiology working spaces in a group with no existing protocol was challenging, and so organization was key. Making sure that the chemicals that I was using were the right ones for the project. So realizing you can't just use any type of grade chemical, you have to, you know, think of chemicals with very low copper concentrations. Of course, consulting with collaborators throughout the time to receive feedback on the project was very important. So moving forward then, once we knew that TITA was not toxic to the organism, the next step was to set up the actual limitation experiment. And so for this purpose, we again did computer modeling, geochemical modeling, to try and estimate the concentrations of copper that we would need to add to the medium to achieve our estimated calculated free copper value. And so for these experiments, we had different copper concentrations, but we also had a positive control, which was the wild type culture or copper replete. And we had a negative control, which had no copper and no TITA. And so shown on this slide are the results of these copper limitation experiments. Again, on the y-axis, we have nitrite concentration and days. And these, again, are a combination of biological replicates and also multiple experiments. And you will see in my next slide how long it took to achieve this beautiful slide here. So the results shown here showed us that there were two cultures where we could achieve copper limitation, and this was estimated to be at 10 to the minus 15 moles per liter of copper-2 that was available to the organism or less. And I also performed an interesting experiment where I tried spiking copper back into this Cu10, which is a copper-limited culture, to see what that would do. And for me, it was really amazing to see that the organism could recover its ability to metabolize ammonia. And so this is this purple line that you have here. And so what that was telling me was that physiologically, this organism could compensate for this limitation if we introduce copper at this particular point in time into the culture. Something else that I was interested in was to know more information about the actual growth of the organism. So these are enzymatic results that we are seeing where we're measuring nitrite output. But I wanted to also see what the growth pattern of the organism looked like. And so for these experiments, I was lucky to collaborate with a researcher in a different department. I had over 100 samples at this point that needed to be analyzed. And luckily, this department had an instrument that could do cell counting. And so I didn't rely on the microscope in this case, which would have taken me a lot of time to try and cell count. But based on the results, so we have cells per mil per culture and number of days, we can see that the cell numbers very much coincide what we were observing in terms of nitrite output. So this actually confirmed that copper was in fact limiting the growth. Copper limitation was in fact limiting the growth of this organism. So then at the very end, we also wanted to be able to relate then the growth rate of the organism to the amount or estimated amounts of copper in the culture. And so as you can see, this actually shows very nicely that there were two cultures that stood out in terms of copper limitation when compared to the copper replete. And so some of the key conclusions from this part of the study was that TITA is not toxic to viennensis, that the copper concentrations that are less than or equal to 10 to the minus 15 mole per liter limit the ammonia oxidation, and that when you reintroduce copper into the cultures, this restores their ability to oxidize ammonia. And it shows that there's a link between copper concentration and the ammonia oxidation activity of this organism. And for us, this is really pivotal because this led then to the transcriptomics part of the project, which I will talk about in a few moments. But before I go there, I wanted to show that as a result of we also addressed some of the competing processes that could be happening in soils that could lead to copper limitation. So we know that can be taken up by microorganisms, but microorganisms have to deal with organic matter binding to copper, dissolved organic matter binding copper, clays and iron oxides binding copper, and so these abiotic reactions can compete with the organisms that are in soil environments that are trying to attain copper. So I'm showing you here again the timeline between the start of the project and basically arriving at these results that I've shown you here, where we could find these limiting conditions. So that took a very, very long time. In between, we could start some of the transcriptomic experiments in parallel so that we could complete the project in time. And in between, I was again lecturing, writing, mentoring. So some of the lessons that I learned during the second phase of the project as I moved forward is that forming collaborations was very, very important to completing the project faster. So the more experts that I had to help with the various steps, the faster we could arrive at our goals. There were also personal life events that tested my resilience to bounce back. So during my postdoc, I lost my dad. And so there was a time there where I really didn't know if I could continue with the postdoc, but again, my family and my friends were there to support me. And so I was able to bounce back and finish the postdoc. There are also difficulties with the experiments that tested my patience as a researcher, because archaea are very hard to cultivate. And as I mentioned before, there were many times where the cultures would crash, and it felt at times like, well, this is over now. But thankfully, you know, with the help of team members, very supportive lab members, I was able to continue with the research. And so that's very important, having the right team. So now we arrive at the transcriptomic part of the project. So now that we had found what the limiting conditions were for this particular organism, naturally, we wanted to know, well, what does that mean in terms of gene expression? And what can that tell us possibly about how this organism can take up copper, how it can store it under these limiting conditions? And so our hypothesis here was that low concentrations of copper would result in the expression of copper uptake and storage genes. So we come to the RNA experiments. So these are really challenging too. But again, I had a really good team. And we could figure out that we needed a lot of biological replicates in order for our transcriptomic results to be statistically significant. And so for us, we chose 8 replicates of the replete and 8 replicates of the copper limiting culture. And along the way, we decided, okay, well, out of the 8, we're going to sacrifice 5 for the sequencing project, and 3 of them we will follow through until its growth stabilizes. So for these experiments, we had to scale up the cultures. This was another challenging part of the project, because we didn't know how the culture would behave when we scaled it up. But thankfully, the growth pattern did not drift away from what we were seeing in the smaller culture we initially started with. And so this contraption that you see at the bottom was something that I engineered in order to carry out the extraction in a very fast but efficient way. And that involved filtering out this growth culture onto a filter. And I'll show you that here. So there's a filter that could be added to the syringe through which the culture could be put through. And once a certain volume went through the filter, I could then quickly take that filter and freeze it. And so the idea being that we would quickly capture the transcription taking place in these different conditions. And there was also having to think about how fast do I need to work between filtering each of the cultures. I should think about how I'm going to take the cultures out of the incubator, and time myself to see how quickly I have to do this filtration procedure. But anyway, following this extraction filtration procedure, I proceeded with the extraction procedure, and this was kind of nerve challenging my nerves as well, because for anyone who's worked with RNA, you also not only have to work quickly, but very carefully to ensure that you don't degrade the material away before you can analyze it. So having successfully done this, then part of the sample was split up to do some quantitative PCR experiments where we would study specifics of interest. And the rest of the sample was then sent out for sequencing. And so another reason for scaling up the experiment was to try and acquire as much RNA as possible from these experiments. So here I kind of summarize what our RNA sequencing and analysis approach was. So for the RNA sequencing, this had to be reverse transcribed, and there were different copies of this cDNA that were then randomly cut into large, equally sized fragments. And we decided to do single reads in this case. We were dealing with an organism where we already knew its proteome from previous studies. And so we were fortunate enough, once we received the sequencing results, we could align those sequences to a reference genome. And being able to then carry out this procedure, we decided to normalize the data to in the number of reads for genes per sample. And we used two approaches for the normalization procedure, in log twofold change. And we used a very low statistical value of 0.01. We visualize the data as shown at the very bottom. So wherever you see red, this is upregulated, and wherever you see blue, this is downregulated. And so upregulated just means that you're expressing that transcript very highly relative to something. And downregulated means that you're expressing that transcript very low relative to something. And so in the following slides, we will show two different ways of visualizing that data between samples. And the rows that you will be looking at will be the different genes. And I will present to you only part of the transcriptomic study, because there are many, many results and many analysis, but here today I will present some of those. So these are the results of the cultivation experiments where we scaled everything up. As you can see, we have copper replete and copper limited conditions. We decided to pick the culture that had the lowest or the most limitation in the initial experiments to do the transcriptomics. And timing also was important here in terms of when we would harvest the replicates to do the sequencing. So I learned that this is very important to decide at which time point you sample for the transcriptomics. You don't want to sample somewhere near the end where the samples could be already going into death phase, but you don't want to sample somewhere where it's too early and cells haven't had a chance to divide. So we chose somewhere in the growth phase where the organism should be dividing. And so, as I mentioned before, we had 5 replicates that we sacrificed for each condition, and then we had 3 which we followed in the limited for the copper replete. One of the cultures did not end up growing, so we only had 2 that we could follow to the end. So now shown here are one of the results of the RNA sequencing. On the left-hand side, you have basically abundances reported as RPKM, and this RPKM is essentially a way of comparing the abundance of reads within a sample to the total number of reads within that sample. So wherever you see replete, that's relative to the genes within that replete sample. But on the right-hand side, you have a ratio reported as log 2 fold count. And this ratio is comparing abundances between the copper limited culture and copper replete. And so among the top 10 upregulated genes that we saw were some interesting multi-copper oxidases. So multi-copper oxidases are a type of protein that in eukaryotes have been shown to oxidize copper. And so this was a very exciting result to see such a gene encoding this protein expressed. There were a lot of unknown and hypothetical proteins as well that looked pretty interesting. Some of them that could possibly contain a copper domain. In addition, we saw permease genes, which are encoding basically proteins that can lie very close to these multibases, and perhaps they can assist in the uptake of copper. And I will talk about this idea a little bit later in the presentation, but we were able to verify these results with qPCR as well as with proteomics on 2 samples with the proteomics. We also observed 10 downregulated genes. A lot of them, again, were hypothetical. We did observe the CDP gene, which encodes this calcium binding protein. And we thought here that this might have something to do with preventing cells from aggregating, perhaps, or it could actually prevent cells from not having an affinity for the copper. So maybe this is a way for the cell to acquire copper as well. Another interesting part of the transcriptomic results that we saw was this ammonia-oxidation pathway. Since we had the physiological results, we knew that the ability of these limited cultures to oxidize ammonia was reduced, and the transcriptome was very nicely matching what we saw in the physiology. So shown here is the electron transport chain for the ammonia oxidation pathway. This AMOB and AMOA, this is the ammonia monooxygenase that carries out the first step in the oxidation procedure. And we see that for the most part parts of this enzyme were down expressed, which would match what we were seeing physiologically. We also saw that there was down expression or down regulation of the ATPase, which is complex 5 and complex 1, and these usually consume protons as part of the ammonia-oxidation pathway, which was really interesting because that would kind of hinted that maybe the cells were trying to preserve proton motophores by shutting down parts of the pathway that would normally consume them. And then to contrast that, we saw certain parts of the transport chain that actually contribute to the proton motophores were turned on. And so another hypothesis that we had as to why is there this difference in components that are on and off as part of this pathway is that perhaps some of the mediators that shuttle electrons, so for example, in complex 3 and complex 4, maybe those are kept on also as a way to shuttle electrons and produce more proton motophores in the event that there is any ammonia that's passing through the AMO and also through this hypothetical protein. Another part of the analysis that we carried out was to compare these copper-related genes between our soil AOA to the marine AOA that was originally studied, and by this time they also had a published study showing the transcriptomic results of this organism. And what was really interesting here was that there was a stark difference in the abundance of copper-related genes that we could see in N viennensis compared to the marine organism. And so that led us to hypothesize that maybe the fact that these two types of AOA inhabit could be a reason why these two lineages or types of AOA have adapted differently to copper uptake in these environments. And so the overall pathway model that we came up with as a result of not only these results, but other results that I didn't have time to present here was, the first copper model for soil-dwelling archaeon, where we used the results of our transcriptomics to predict possible ways that copper could be taken up by the organism, some involving these multi-copper oxidases, others involving genes that, or proteins that are normally considered as part of copper resistance, but have since been shown that they could be possibly involved in copper uptake. So the model itself came about through comparison with data from other studies in which these various proteins were being hinted at to possibly be involved in copper uptake. And also the redox state of the copper came from information from those studies, which talked about the type of copper that was being bound by these different proteins. So the key conclusions then to transcriptomic study was that copper limitation triggers the upregulation of copper-related genes, including those that could possibly be involved in copper uptake and storage. Copper limitation also caused the downregulation of certain genes that were involved in the ammonia-oxidation pathway as a way to conserve energy. And compared to copper-related genes in the marine AOA, N viennensis appears to respond differently to copper limitation, suggesting that they may use different copper uptake pathways. And so what this means overall, as far as nitrogen cycling is concerned in the environment, is that there could be environments in soils and in peat bogs in which the concentrations of copper, what is bioavailable to the organism is low enough that it would inhibit this ammonia oxidation pathway. And perhaps in these limited environments, these could be ideal places to copper acquisition pathways. Now that we have sort of hints and possibilities as to what those systems could be, naturally one of the next steps that is being planned as part of this project is to look at different kinds of environments to search for possible presence of these pathways that we've now hypothesized as part of the cultivation work. And so once again, to give you a sense of the time as far as this project goes, just because the project ended, the project didn't actually end in terms of writing the publications, teaching responsibilities, networking, and finding what to do next. But thankfully, as of February, we were able to publish the first results. And as of early this week, we were able to publish our transcriptomic results. And so you can find the results from the first half of the study in Research in Microbiology. The second part of the project in the EastMed Journal, which was published this week. And so with that, I'd like to end the presentation by thanking all my co-authors, in particular Logan Hotchkiss, who's a PhD student who did all the transcriptomic analysis. So he was very instrumental in producing these results. But there were many, many people that were involved along the way in terms of providing input to the project and also technical support. And of course, I have to thank the European Union for providing funding for the project, and also to the Austrian FWF funding agency for providing funding along the way. So I will take questions now, which can be on anything in the presentation.
Thank you so much, Carolina.
You're welcome.
Bringing my video back on. There we go. Anybody, this can be questions about the science, questions about Carolina's path.
I had a question. I was just, so there was a part, and I can't remember exact significance in, I believe it was an activation. But you said you were using a p-value of smaller than 0.01. Is there a specific reason why your significance value was so low? I know traditionally it's 0.05 in different fields, but if you could just kind of elaborate more on why your significance threshold was so small.
Yes, decided on this because there were a lot of transcripts that have been shown in other archaeal studies where significance in terms of the p-value, but if you were to only look at the value, you would overlook them. If you were just basing the whole analysis on log2 fold differences. So we decided on this value, what was known at the time.
Cool, thank you.
Emily has a question.
Hi. So my question was towards the beginning of your presentation. You had mentioned how you dealt with a lot of adversity, and just against students about you don't belong here and like X, Y, and Z. And I just wanted to know how you overcame that with so many people telling you where you do belong, quote unquote, and what kept you, staying where you were and believing that you did belong.
Yes, well, throughout this, these encounters, of course, naturally there was a lot of anger and sadness, at times disappointment, that people could think that, you know, in such a way. But what helped was actually being able to discuss this topic, not only with friends, but with my family, because I have a lot of family members that have experienced a similar situation, and trying to stand above that, knowing that none of it was true. I didn't believe it, you know, that I wasn't smart enough to be where I was. When strangers would say, you know, go back to your country, and first thoughts were, well, you should go back to school and become more educated. So I think a lot of it had to do with confidence in myself, and also because I had so many loving and supporting people that knew none of this to be true, and who could give me positive reinforcement regarding that.
Wow, thank you. Thank you for sharing.
That's such a great answer, Carolina. I just want to underscore the importance of community and family. Yeah, I think that a lot of times, especially when students are first enough to kind of distance themselves from their background or who they are, that they have to change somehow, and it's actually who you are that makes you strong, and those ties and those bonds to community.
Yeah, for sure. I mean, being able to have these discussions with family members who might have experienced the same, especially my parents, especially my dad. I know when I arrived to the US, he had a lot of these experiences. Yeah, and being able to talk to my dad about that, and talk to, yeah, cousins and aunts and whatnot.
Were you able to find support in your academic community as well?
Yeah, for sure. So at UCLA, as I mentioned, I was part of this, and a lot of students were students of color in the program. So I could also, you know, lean on them for support, and talk about my experience and hear about their own experience. So definitely being a part of these STEM programs has always been pivotal. Even to this day, I still remain active as part of the SACNES community. And yeah, I love it. I love seeing and being able to relate. And I've actually had student major conferences, especially the geochemical ones, that have come up to me and are like, wow, you know, you're one of the very few people of color that I've seen give a keynote talk, or, you know, how do you do it? Or, you know, you're the first person that I can talk to that looks the same as me. And so I think it's, yeah, also being a role model to people has also been important.
That helps a lot, because also first generation, and sometimes I want to apply to Stanford for my PhD, you know, graduate program, and I'm just afraid because I've that imposter syndrome, you know, and so I'm really trying to remember, like, I am enough and I'm smart, but sometimes that gets the best of me, because even description they put, we want the best students, and I'm like, what if I'm not one of the best students?
Oh boy. I mean, I had that experience too, where I had some people say, well, I went to Stanford, or I went to Harvard, and you went to what school, and you know, and just, you know, just laughing it and being like, yeah, okay, so that doesn't cancel any of my success, or, you know, it doesn't cancel anything that I've done.
Yes, it cut off the last part. Thank you.
Great question, Emily. Thank you so much. Anybody else? Other questions? You have a great opportunity here. Don't squander it. Take advantage of the fact that we got Carolina live from Austria.
Average, how much did you spend on in lab, like doing your lab work?
Woo, okay, that's it.
Good question. Yeah, work-life balance.
Yes. So as a PhD student, I can tell you I was there on weekends.
That's how she landed those 2 big grants, those 2 postdoc positions.
Yeah, I was there on weekends, but I was very fortunate enough to be living on campus at the time, even though it was quite then, but it allowed me the flexibility of being able to go to the lab, you know, at all hours when I needed to, which was pretty much the whole time. But after that, during my postdoc, I don't know if I became, I guess I became better. I don't know, working more efficiently, becoming more experienced, but I found that I didn't have to go into the lab on weekends. And actually, in the town that I was at, the university that I was in, they did not allow students to go in on the weekends. So you had to complete your work within the work week. And so there it was all about managing my time so that I could complete everything within the working time. So that meant not going in on the weekends, and also being in this culture. One of I guess cultural shocks for me was how more relaxed the people were in terms of holidays. So there are a lot of holidays, at least 5 weeks worth of holidays are a requirement for everyone. So you have to, and it was so weird to have my host basically say, okay, well, you know, you have to use up your holidays. So there's a shift also in work mentality, living in this different environment. And then for my second postdoc, by then I pretty much had it all together, I guess, in terms of getting all my work done within the work week. And there it was quite easy to time manage between time I would spend in the lab, which never past 6, and that's what I told myself going in. You know, I'm never going to be here past 6 o'clock. I'm going to make sure early enough to finish what I have to finish, because I want to go home at the end of the day and have a life. So yeah, I think all of this time management and work life came about just over the years, as experience, getting at working more efficiently in the lab and managing my time.
Thank
you. You're welcome.
So Carolina, I have a questions for you. So how do you retain all the information that you have learned?
How do I retain it? I think part of it has been to continue to bridge not only information and, you know, as far as methods go, but topic-wise, I feel like there's an overlying theme or underlying theme behind all the projects, and this has been metal cycling and how these organisms play a role in that in the environment. So I think part of the way in which I retain all this information is connecting all the different topics that I have. And actually, Chad mentioned I will be starting a new position next month in Switzerland, looking at biobatteries and fungi. And you would think, well, that doesn't sound like anything related to what she's done so far, but actually it interconnects my PhD project, in which I was looking at redox processes in a bacterium and learning about its chain, And now applying that knowledge to build a biobattery, because actually components of that work in the same way that you would find them in this model system that I studied as a PhD student. So I think part of obtaining all this information is building this overlap projects, but something huge that has helped has been teaching and lecturing on different topics, and oftentimes on topics that I've never done research about, but I needed to papers, mostly on asking questions whenever I didn't understand something. So that was also told at the very beginning, admitting, I don't actually know this topic in depth, but having to learn it in such a way that I could teach someone has helped, I think, in being able to retain all that information. So teaching is very vital, I think.
Thank you.
You're welcome.
Hi, other question. I just wanted to know, as a PhD student, how would you advise an upcoming or an aspiring PhD student to, oh, can you hear me?
Yeah, I can hear.
Sorry, my internet's really.
Do you want to type your question into the chat box, Emily? She might have lost internet for a second there. Well, I'm going to follow up on that, and hopefully Emily's going to be right back with us. So actually, we just had a couple of workshops about applying to grad school. We had a GRFP workshop to to kind of look at the structure of applying for that fellowship, and then we did an overview. So a lot of our students right now are thinking about grad school and trying to organize, the project of applying and the timeline. And so would you have advice about that? Like, how did, writing the personal stuff, how did you plan for that and organize that?
Yeah, the part of the organization is starting early to research all the information, give enough time, and that goes for all kinds of applications throughout your career as a scientist. Not only for grad applications, but later for postdocs, for professorships. It's always important to prepare yourself ahead of time and ask people questions. Don't be shy about asking even the people within the department. I mean, that was not something that I learned about until later on. Like, it's okay to reach out to people that will potentially be a part of your group, to ask, what their overall experience was working in the department or for a particular lab. Yeah, that's really one of the most essential to do, to gather all this info so that you have enough time and you're not rushing at the very end. As far as like getting letters of recommendation, yeah, you also need, the time in order to give your letter writers enough time to prepare the material for you, and reminding them, because you're not the only thing on their agenda, so you need to remind them, this is coming up, I'm just letting you know, like, it's 1 month before and 2 weeks before, it's a few days before.
That's right. Those reminders are important, yes.
Yeah,
it looks go ahead.
Go ahead.
I was just going to say, it looks like Emily's back, so I just wanted to encourage Emily to let us to say her question. I just kind of framed it in terms of a general grad school preparation advice question, but we'd love to hear what your question was, Emily.
Thank you. I'm so sorry about that. This has been happening the entire Zoom call. But I was going to ask how a PhD student manages their ideas and their thoughts, because I know for me, I have so many ideas, but I know in grad school, I'm sure you probably only focus on, right, and you do research on like one big idea you have and then kind of go with that after you graduate. But how do you manage all that, all the ideas?
Yeah, well, yes, I had many ideas as well. As Chad would know. And sometimes, you know, it's actually good, I would say, to go with some of those ideas off tangent, even if they are not the main focus of your project. I think in science we call those like our windowsill experiments, where we can work on them on the side in addition to our main project. And actually I would encourage that, because sometimes it's the side projects that become, in the end, depending on how juicy they are, coming from that. So I would always encourage you to have, you know, to follow through with those ideas, and also to keep a notebook of all those ideas as well, because it's something you can come back to as you're thinking later on, what kind of projects can I write for a future fellowship or for a future position. You can always refer back to your.
Sorry, I was muted, but thank you.
You're welcome.
For keeping that in mind.
Yeah, I had a lot of those side projects throughout my postdocs. Some which I was hoping and actually did end up using as preliminary work on proposals. Some of those I put aside, literally put aside in a drawer as potential, you know, like future projects. And there's ideas where I've put together posters, and I've gone to say like a course that I've been invited to, and I've presented those ideas to experts in that particular field to kind of get a sense, okay, is this a good idea? Is this a bad idea? And that's also helped fuel like additional plans as to take things.
I have a question. First of all, thank you for the great presentation. And second, have you ever, how do I reframe this? What is your ritual, if you have one, where you to do when you feel like you don't belong or you're out of place in a group, or just in STEM?
I see. What do I do? My ritual? Well, definitely calmly, and there's actually my first, the first address where I go to when these insecurities or these situations arise. Always calling my mom or my niece and talking through it, because yeah, I mean they act as mirrors, and also they're able to deny those insecurities and remind me of how far I've come. My second, of course, address are a lot of close colleagues that are also part of STEM. I mentioned that I have met a lot of people through my STEM program, so also reaching out to them, because I know, you know, they have reached out to me as well during times where they felt insecure, or they've been going through hard moments. So I encourage you to keep in touch with all you're meeting right now as a part of your STEM programs, because I feel like it's, as I said, very important to have this sense of community to hold you up in times where you may feel like, oh, I don't belong, or what am I doing here? To remind you, hey, you do belong, of course, and you're not alone.
Love it. Thank you so much.
You're welcome.
When you're undergoing a difficult situation, how do you like maintain motivated? because what happens, I tend to lose a lot of motivation.
Yeah, I've had moments like that too. Throughout my postdoc, especially when all those things were going wrong. I have to take a moment to, I guess, meditate in a way and think through all the times that I succeeded, despite all the difficulties that came up, and think through specific moments where I was challenged like that. That really helps. But also talking it out. I like socializing a lot. I'm a very social person. So I talk to my husband a lot, and he'll be like, "What are you talking about? You're amazing." What is this insecurity? Why is it coming up? Why are you questioning things? So having that support system in place, but also taking a moment and thinking through, hey, wait a second, let's not forget, X, Y, and Z that I've done. And so this too shall pass.
Thank you.
You're welcome.
Great question so far. Any additional questions? No other questions?
I was wondering, how was that you did the process of applying to make the jump from being a student in the US to being a student in Europe, and was it hard to get accepted?
Yeah, so to make the jump, that's starting to gather information myself for this, so I had to have some idea of where abroad I wanted to go, and for me that was Germany. And starting to reach out to the network that I had built up. For example, people that I met through the geobiology program, people that I had heard in conferences. Attending conferences is really important because that's where you will meet a lot of potential people that you will come to work with. So I not only attended conferences in the US, but during my PhD, I went abroad for conferences. And so by preparing myself really in advance, I mean like 2 years ahead of time, looking at what kind of fellowship opportunities are there to go abroad? What are the requirements? So for example, for Fulbright, they had an initial language requirement that I found out about. And so for that, in order to get a recommendation from a professor teaching German, I enrolled in a German course for a year during my PhD to start not only gaining language, but also I wanted this person to recommend me at the end. So I wanted to do well in the class. And so that played a pivotal role. And as I mentioned before, keeping these ideas that would come up throughout the PhD inform eventually the proposal. So you have to give yourself enough time to do all this.
To the point that we've been making about getting started early, right? And having time to do that research and really think through things. Good advice. Other questions?
Thank you.
You're welcome.
So, Caro, this is more of a comment, but in all my time that I got to interact with you, one of the things that I really saw steady in your sort of personality is how positive you have always been. And so no matter what struggle you face, you always maintain the positivity in dealing with it, navigating it, journey. And of course, the magical laugh that's contagious. So how do you do this? How do you face obstacles and what do you do to stay motivated and positive? Like when you're facing an obstacle, how do you stay positive about it?
Yeah, well, part of that positivity has having other positive people surround me and surrounding myself with people that are similar theme, like they have goals, or they have very strong personalities, as far as, not letting dire circumstances hold them down. So one of those people that has been really important, positively in my life, has been my aunt, who lives in Mexico, and her name is Marta, or Tia Marta. And oh, I was positive. I mean, she is like, she survived, a car accident where she was paralyzed. She would ever recover movement. And you look at her having survived that, and how positive and cheerful she is. And she's had other circumstances happen to her where I was just like, I don't know if I would have remained positive in her situation, but having these role models in my life and surrounding myself by people that think positively, have positive energy, really important for keeping me motivated and keeping me striving for my goals and my dreams. And my sister was also that way. She had a very strong personality, and even if you're like, well, you're wrong about this, she could argue that. Like, no, this is my belief, and you can tell me I'm sticking to what I believe in. So having a lot of strong role models.
Thank you.
Carolina, I think you're going to be a strong role model for most of our students who got to hear your talk today.
I hope so. I haven't given up my dream of becoming a professor someday.
We can't wait to get you in the university classroom.
I've been saying that since grad school. Someday.
Well, we've seen you fulfill all your other goals, so we don't have any doubt that you'll make that one too.
Yeah, I'll make it. Slowly but surely, or maybe fast, but I'll make it there.
Any last comments or questions? We're coming at the end of a second hour here, so I want to be respectful of Carolina's time. It's getting late back in Austria. Probably what, 7, 8 o'clock now?
Almost 8.
Yeah. Okay.
I have a question actually.
Okay, Claudia.
First, Carolina, thank you for the presentation. I really enjoy it. I think I learned a lot. My question is, what would your advice be for a first generation college student from an underrepresented background who's trying to go into academia as a I don't know how to phrase this question. Yeah, what would your advice be for someone who's trying to go into academia, who never really, with a lot of limitations? Like, what would you tell them?
Yeah, I would tell them, first of all, start building up your network, your scientific network, your professional network of people, because these people will be really important for you later on as you start to build up your academic career. And the more people that you know and that you start to include in your circle, the more opportunities you'll learn about as well that you can take advantage of. And I would say, even if you're afraid of going for certain opportunities, whether that be volunteering to present more often, for example, for the STEM program, or taking the risk of doing oral presentations at conferences where you may not feel comfortable, I would say do it, because that also gives you exposure to the scientific community. People start to know who you are. If you're just hiding somewhere in the background because you're shy, no one's going to notice. But if you put yourself in those situations, even if they're uncomfortable, I think in the end, they can also help people know more about you. So as someone coming from a first generation university student, that's something that I've learned along the way. I mean, I was so terrified at giving even like the simplest talk among colleagues. I mean, Chad even knows this. I was so terrified to even talk to a small group of people, and to go from that to now talking to you, an audience of 60 plus participants.
You had 88, an audience of 88 at one point today.
All right, 88 participants has been a long journey, but I'm glad that I continue to put myself out there, and despite being terrified and feeling like I'm the shyest person ever, doing it because people started to know, who I was, and I could start to network with a lot more people.
Thank you.
You're welcome.
That's a great answer. Feel the fear and do it anyway.
Yeah.
Okay, well, I think we've reached the end of today's presentation. I want to thank you again so much, Carolina. It's been so great to spend this time with you, and we really appreciate you making the time to talk to our scholars and to our community.
You're welcome.
Go ahead.
I said you're welcome. I'm really, really happy that I had this opportunity to talk to all the students, and of course to see my faculty, previous faculty. Again, Karen, I see you there.
and our plan was, Yuli and I were going to bring Carolina here in person before everything happened with COVID. So that is still a future aspiration. So hopefully, maybe even next year, we can make it happen that you can get to come back to campus in person. And I hope everybody that's here today will come to that talk as well.
Yeah, I would love that opportunity.
Yeah, we would love to have you back.
Yeah, we look forward to hosting you in person for sure.
Absolutely. Okay. Well, thank you again so much. Go have a nice dinner. Have a great evening. We'll be in touch, and we'll look forward to bringing you out here once everything gets back to normal. Thank you, everybody, for coming today.
Thank you, Carolina.
We'll see you at next week. We have
you.
another talk, and we'll send out emails.
Thank you.
Thank you.
Thank you.
Okay. Thank
Thank
you.
you.
Thank you.
Thanks.